Thank you very much. First of all, I would like to thank the organizers for organizing this meeting and for inviting me. I should say right from the start that I'm neither a geneticist nor a historian, so before you begin wondering what I'm doing here, I'm very much interested in the public representation of science, especially when it comes to science education. And I find very problematic the romantic, naive hero worshipping that we often find in textbooks, because it distorts how science is done. And this is why perhaps my presentation will be a bit annoying for people who like this perspective. So I was asked to talk about the Mendelian gene, and here is an overview of my talk. I will begin with a distinction which is crucial, especially for science education, the public understanding of science. When we're talking about the Mendelian gene, are we talking about Mendel's gene or the gene of Mendelian genetics? Then I'm going to argue that the first doesn't exist. There's no such thing as Mendel's gene. And my main point would be that the stereotypical account of Mendelian genetics, what some people previously described as the orthodox, distorts both science and history. And I'm going to conclude with some suggestions as what to do with genetics education. So here is a way to distinguish between the two. Mendel's gene is something that some people have inferred from his own paper. The elementa or the factors, Stefan and Kirsten described that. I don't have to say anything more about this. Whereas what we know as the gene of Mendelian genetics was something that was developed later. The representation you see here comes from the book by Morgan and colleagues, where genes are represented as beads on a string. As already been told, the concept itself was coined by Wilhelm Johansen, and he explained that there was no need to specify what a gene was. He discussed in the paper the previous attempts to come up with units of heredity in previous theories during the latter 19th century, and he proposed the definition that we see here. Then we have this fabulous, enormous program on Drosophila by Thomas Han Margan and his colleagues and students. Initially, they didn't use the word gene, but in the end, they adopted the term. And this is again the representation that they used of genes on chromosomes as beads on strings. As you know, they were able to map genes and establish the program that we call classical genetics. Now, the most important thing is that whereas we see in their work talking about genes for a trait, they explain in this book they wrote in 1915 that when we're talking about a gene for a trait, we're talking about one of many genes that contribute to a trait, which when changes might bring about a change in the phenotype. In other words, they accept and explain that there are many genes contributing to traits, to a single trait, yet it is possible if one of these gene changes to have a change in the phenotype. This is 1915. Unfortunately, even today, we still teach our students in schools that there are unique genes that control specific traits. So what they introduced there is the idea of a gene as a difference maker. Here is an illustration of this. We can have up to 25 genes contributing to the color of the eye in Drosophila. If one of these changes, we can have a change in the color. In this sense, gene 3 in this case is a difference maker. It is a cause that brings about an effect, but it is not the only causal factor for the development of the trait. In 1933, Morgan receives the Nobel Prize in Physiology and Medicine for his work on genetics, and he writes in his Nobel lecture that at the time there was no consensus of opinion among geneticists as to where genes were, if they were real or purely fictitious, and he famously pointed out that it doesn't make a difference if they're real or not, because the concept itself had a great heuristic value for research. So the gene of Mendelian genetics, we can describe, and this is the presentation we find in textbooks and in the public sphere, a factor that controls a trait. It was not specific about what that was. It came, the molecular gene came up later, and this is what we usually describe as the classical gene concept or the gene of classical genetics. Now, the question is, did Mendel come up with this, as we usually see in textbooks and everywhere? Here is something from a page from the educational section of Nature, the prestigious journal Nature, I must note that they acknowledge on the page that it's not been updated. Yet, if a student looks about what Mendel did and what genes are on that page, she will read that Mendel hypothesized that each parent contributes some particulate matter to the offspring. He called this heritable substance elemental. Or if we go to Encyclopedia, we see that Mendel's genes were only hypothetical entities, factors that could be inferred to exist in order to explain his results. Now, the question is, did he really do that? If we read the paper, and I'm following here the excellent translation by Staffan and Curtin, we see all the time, I will not go to the details of the quotations, that he's talking about traits, he's talking about characters. And he's talking about dominating and recessive characters all the time. And his aim was to see what happens when we cross hybrids and how these traits are transmitted from one generation to the next. The important thing here is that he mentions what has been translated as hybrid traits. He's not talking about the traits of the hybrids. He's not talking about traits which are themselves hybrids, and he explains in detail what he means. And in the end, he clearly shows that there are traits which are dominating. These are the ones that come from the parental generation, and others which are latent and may reappear in the next generation. He is talking all the time about traits and not factors or elements or whatever. And he ends up with ratios that resemble the laws that we know, the segregation and independent assortment, even though he does not describe these as laws. I think that the most interesting part is this one. At some point, he has results that resemble the 9:3 to 3:1 ratio, but he does not describe that in this way. Rather, he's using this description. He's talking about 9 members, and he describes what is going on in each case. So the experimental result that he presents on that page is 315 round and yellow, 101 angular yellow, 108 round green, and 32 angular green. And he goes on to represent these as different classes on the basis of which phenotypic traits, phenotypic characters they have. And he ends up with the ratio that you see here. This is very different from the representation that we would give today. The modern ratios, if A capital and small a were alleles, we would be talking about the classic 9:3 to 3:1 ratio. So at first point, he does not describe things in the way modern or the approach of Mendelian genetics would have things. And while wondering why, I came across Robert Olby's classic paper, and then the book that has this paper in the appendix, the 1985 book, in which he elegantly explains why he did that and how we come up with this representation. So Olby has a table like this one in his book, Combining Phenotypes, and he explains how Mendel combined the two characters together and ended up with this ratio. Whereas I have put below what we would do today, in classical representation of Mendelian genetics, and why, if we thought of A and A, big A, small a, in terms of alleles, we would end up with a classic 9:3:1 ratio. But because Mendel did not think of big A and small a as alleles, but as phenotypes, he ended up with a different ratio. So in my view, that's the most convincing element that Mendel was not thinking in terms of alleles, independently of what we say and what we see in textbook. And I think that Bob Olby gives the most convincing argument for this. So in other words, Mendel's terminology was about dominating and recessive traits, and big A, small a was the hybrid form. Whereas in modern terminology, we know that each letter corresponds to an allele, and then we have homozygotes and heterozygotes. very different things. So if we read carefully Mendel's paper and the historians who have studied Mendel, we can arrive at some broad conclusions about his paper. He intended to study hybridization in particular and not heredity in general. He studied how traits were passed on from one generation to the next one, and nowhere did he write about hereditary particles that determine these traits. Staffan this morning explained what he meant by factoren, and the comments they have in the translation are amazingly clear. And he was able to observe the segregation and independent assortment of traits, but he did not conclude anything about the segregation and independent assortment of alleles. In other words, he observed what would happen, the result of segregation and independent assortment of alleles, but he did not talk about this. He just documented the finding. And returning to Bob Olby, in his paper and book, he writes that if we think of Mendel as someone who thought in terms of hereditary particles, he was not a Mendelian. But if we think of him as someone who thought of hereditary transmission in terms of independent character pairs and statistical relations, yes, he was a Mendelian. And I think that this excerpt represents in a magnificent way what happened before 1900 and after 1900. Mendel's extremely ingenious experimental approach was adopted in a different context in which it made sense and it was very useful. But this is not what exactly he had in mind when he did the research. Now, to the point that is of most interest to me, why the stereotypical account of Mendelian genetics distorts both science and history. We teach students that there are single alleles that control traits that are either green or yellow, either round or wrinkled and whatever. But already in 1902, it was clear that things are not that simple. And I don't know, I saw the discussion earlier about whether this is the seed or the seed code or whatever. I think the key point in this image is that there's a continuum of traits. It's not either yellow or green. It's a continuum from greenish, yellow to yellowish green, or whatever color this is. I know that the seed code colors are different. I do not know if this is the original color, if this is colorized. We have to ask Greg Ragdig about this. But I think the main idea here is that there's a continuum of colors. And we shouldn't forget that Mendel artificially arrived at purebred varieties, which means, in simple sense, at least to me, that he eliminated the variation that naturally exists in nature. I think we should make clear the distinction between an experimental system in which we can arrive at phenotypes we want to study and the natural variation that exists in nature. So it is also well known, because we discussed this, that even Bateson had found exceptions, and they tried with auxiliary hypotheses to accommodate them in what was to become Mendelian genetics. And I very much urge you to read this book, even if you don't like the cover, because it shows in an elegant manner why the genetics of the 20th century became the Mendelian genetics that we know, and what could have happened if it was Weldon who would have dominated the field. Now, my problem is that this simplistic, naive account that we teach, Adam Rutherford mentioned that yesterday, you know, blue is recessive, dark brown is dominant and blue is recessive. It is an oversimplification that deprives students from realizing the enormous variation that actually exists in nature. And it is interesting, at least this is the study I have found, already in 1909, there have been findings that show that it was possible for two parents with blue eyes to have a child with brown eyes. So I think it is deeply problematic when we teach students at schools, that's my main concern, universities are different, that there is one gene or one allele for one trait. And the naive understanding that emerges from that, this is another excellent book you should read, can be very harmful. And Amir Teicher shows in this book how Mendelian genetics was co-opted by the Nazis to develop their policies and eventually their eugenic policies and their eugenic programs, and how they actually taught Mendelian genetics in schools to convince people about what they were doing. My concern of today, I have written in my own book about a concept that I see everywhere, which is again what Adam Rutherford showed yesterday with the media headlines, what I call the associated gene. Because people are taught in Mendelian genetics in schools that they're genes for traits, and because they are not given the full view, sometimes they confuse association with causation. And they read in the headlines that, you know, gene for that was found, whereas what was actually found in the study, if you read the study, is that an association was found, and we need to explore that further. And we have so many GWAS these days that find many interesting, more or less useful correlations. But the big problem is, as every geneticist knows, is that insofar as we understand the underlying biological mechanism, there's not much we can do, because a correlation can mean that two genes contribute to a condition, or that one gene controls the expression of the other that contributes to the condition, or that both control the expression of a third gene that contributes to the condition. So associations can be useful, but not very useful until we understand what is going on. So what we should be showing students in schools is that we have pleiotropic genes and polygenic traits, and that the relationship between genes and traits, or alleles and traits, is a many-to-many one, and not a one-to-one, as the stereotypical teaching of Mendelian genetics shows. And of course, this has nothing to do with Mendel. This has to do with how we explain science to students. The second problem is that the lonely monk, actually he was a friar, as the people explained yesterday, in the garden, this is a huge distortion of how science is done. Mendel was not a lonely genius ahead of his time working in an isolated place. He was very much a man of his time. He was an ingenious breeder, a person who developed a very useful experimental approach that was co-opted later. But this is something that we need to explain in teaching. So if you look at history, during the time since the publication of The Origin of Species, many people were trying to develop theories of heredity, including William Brooks, Herbert Spencer, Francis Galton, August Weismann, Hugo de Vries, and Carl Volongelli, with whom Mendel had a brief correspondence. Mendel is an outsider in this picture, perhaps because he was doing hybridization and people either did not read his work, or if they read it, as Nageli did, thought that it was a special case. So we should stop showing the big white men who are geniuses in science and represent science as a lonely endeavor, and explain to students that science is a social process. And of course, things are not really that bad, as you can see here, but everyone who has published the paper or went to a conference knows that things are not easy. Yet the important issue is that it is the scientific community that decides and promotes advances, not the knowledge that we have. It's not individuals, and everyone has always depended on someone before them, if you read the history of science. Regarding the rediscovery that some people talked about yesterday, there's an interesting case in Augustine Branigan's paper, and then 2 years later, the book, The Social Base of Scientific Discoveries, in which he explains briefly that Hugo de Vries was familiar with Mendel's work, but perhaps he did not think of that as very important. He did not mention Mendel in his first paper, published in 1900, and then Karl Korenz made sure to give priority to Mendel in order to resolve a potential priority dispute between himself and Hugo de Vries. Eventually, in the second paper that de Vries wrote, he mentioned Mendel. But it's not, it seems, or at least following Branigan's and I think Olby's analysis, that Hugo de Vries wanted to let Mendel go. It is just that he thought differently about his work. What is most important is that Francis Galton and August Weismann and all the research in cytology that had emerged since 1865 had created a new framework what has been described as hard heredity. And when Mendel's paper was reread or read in that new context, it made sense in a different way. And this is when it was co-opted and used, and it became, and it should be considered as the foundational document of genetics. So what to do then in genetics education? I recently wrote a chapter in this book called asking the question if we should give peace a chance. And my answer is no, we should not. We should get Mendel and Mendelian genetics out of the biology curricula in schools. Because I think, from my experience as a teacher and a teacher educator, that it does more harm than good. I think we should stop making simplistic connections between genotypes and phenotypes as we do in textbooks, and give students a sense of what development is. And I like this representation by Massimo Pigliucci that shows how our understanding advanced during the 20th century and how we ended up realizing that it's not just about genes or just about DNA. There's a lot more, and there's an interaction that we cannot really separate depending on what goes on in development. So just to show you what I think we should be doing in schools, we should replace this simplistic account that once you have the DNA, then you have the phenotype. Once you have a specific allele, you have a phenotype, with a different developmental approach that shows that the same DNA into different developmental systems. And as you're in the UK, I know you know the chef on the left, perhaps you don't know the other on the right. The same DNA into different developmental systems can give different outcomes, and I have empirical evidence for both of these outcomes. And I think this is a very simple way. That's a metaphor that has problems. All metaphors have problems. But I think compared to the previous one, it gives students an idea that it's not all about DNA, it's not all about genes. It's also about interactions between genes, genes that are in the environment, cells in the environment, and a lot more. Now, if we want to teach about Mendel, here's a great case for the nature of science based on history of science. This is an analysis we did some years ago in textbooks. And we found, based on a specific framework about NOS aspects, as you can hear, we just analyzed the sections of Mendelian genetics of some textbooks, and we found 237 instances in which teachers could talk about how science is done. They were all implicit. And regarding to the aspects of scientific inquiry, we did the same analysis, and we found 125 implicit instances and only 3 explicit. So Mendel gives us a great opportunity to talk about how science is done, but we rarely do this because this is not what we present in textbooks. To summarize, I think that Mendel was a genius plant breeder of his time, not a lonely genius ahead of his time who understood heredity. And I also think that Mendelian genetics taught in schools, again, I emphasize, I'm talking about schools and the education that future citizens will receive, provide oversimplified and over inaccurate explanations for hereditary phenomena. So my suggestion is that perhaps we should drop Mendelian genetics altogether from genetics curricula in schools. If we want to teach about Mendel, we should teach about what he did and why he did it in its own context and not present his work in anachronistic terms. I think that Mendel's story can provide useful lessons for the nature of science, but not for today's genetics. And with this, I'd like to thank you for your attention, and I'm happy to answer any questions that you may have.